Hello, everyone. Welcome to the Science Cafe, streaming live from the beautiful Verde Valley here in the heart of north central Arizona. I'm Drew Cannon. Briefly, before we begin this evening, for those who aren't familiar with the concept of a Science Cafe, it's kind of a movement springing up in communities worldwide where individuals whose field of study and careers that are related to STEM, an acronym that stands for Science, Technology, Engineering, and Mathematics, they meet with you and me, the public, in an informal setting outside of their labs or classrooms in order to just have an informal conversation about their areas of expertise. Our Science Cafe here in the Verde Valley is hosted by the Verde Valley STREAM Council, which is part of the Rural Activation Innovation Network, also known as RAIN, and is made possible through a grant from the National Science Foundation. Their mission is to develop and support partnerships that help shift cultural awareness to target and enhance informal STEM learning for all ages here in our rural community, right here in our home, the Verde Valley. Before COVID-19, these sessions might meet in person in a relaxed environment, such as at a coffee shop, a restaurant, or perhaps a wine tasting room. But for now, anyway, we're doing it virtually from the comfort of our homes. So go ahead and relax, grab a beverage or snack, and let's enjoy an evening of discovery. We do want to encourage you to ask questions. We hope you'll have many this evening, and this is how it will work. As you think of your questions during our guest presentation, please use the Zoom meeting Q&A box to enter your questions, and I'll relay them to our guest after his presentation. So let's talk about our topic this evening, the virus that causes the COVID-19 disease and the vaccines that can help control the spread. We decided to talk about this subject because there's a lot of discussion, but not much of an opportunity to delve into the details, the anatomy of the virus and the vaccines, the science, and to be able to ask the questions we have in our minds. When we choose these topics for our Science Cafe, we try to make it relevant and of interest to our local community. Here in Arizona, the COVID-19 vaccine eligibility was open to every approved age group for those who would like to receive the vaccine. And there are those in our community who have received one of the vaccines, others who have decided they never will, and those who may have questions. But whoever you are, we hope you'll learn something you didn't know before. Our guest this evening is Dr. Matt Pearcy. Dr. Pearcy earned a Bachelor of Science degree in molecular biology and biochemistry from the University of Idaho. He earned his PhD from the Center for Infectious Disease and Vaccinology at Arizona State University, and he currently teaches microbiology, anatomy, and physiology, molecular genetics, and virology at Yavapai College Verde Valley campus right here in Clarkdale, Arizona. And Matt, I understand you're also a river guide at the Grand Canyon during the summer months. Sounds like you're living the life, and I hope I covered your introduction correctly. So welcome, Matt, and thank you for joining us this evening. And briefly before we begin, I wanted to mention I recently had the privilege of hearing Matt's presentation, and I thought it was one of the best I've experienced on the subject. Matt explains the subject in a way that dives into the science and explains it in a way that is understandable. It answered many of my questions, and I personally had a few aha moments, and it's our hope that it will do the same for you. And this is one of the reasons why Matt is considered one of the best of many excellent professors teaching at Yavapai College. First, we'll begin with Matt's presentation. It's about 70 minutes in length, and then we'll have time for Q&A. So without further ado, Matt, shall we begin with your presentation?
All right, Drew, thank you for that introduction. I better be on it tonight. I feel like I have something to live up to now. So, and everybody else out there, thank you for coming out on a Friday afternoon / evening to talk a little bit about virology and vaccinology. So I'm going to share my screen. So what I want to do is just tell a story about viruses in general. We really need to put those in the context of a lot of other kinds of organisms that can infect us and cause all kinds of problems. And after we talk about that just for a minute, then we'll talk specifically about coronaviruses, how many there are, where they've been in the story of humans, how long they've been there, some of the other viruses that have popped up that are coronaviruses. And then we'll talk a little bit of cell biology, how the virus is interacting with our body, with our cells. And then we'll talk about how vaccines are made, specifically the mRNA vaccines, which I think is just kind of groundbreaking and fascinating technology. And then at the very end, we'll talk about the process that those vaccines go through before they're allowed to be, in widespread distribution, worldwide. And we'll look at a specific example. We'll look at the actual data for the Moderna vaccine as it went through all of those processes. And then at the end of it, any questions anyone wants to have, be more than happy to attempt to answer them. So to get started on this, the first thing I want to talk about is these words here, right? Just to make sure we're all on the same page. So COVID-19 is coronavirus disease 2019. So COVID-19 is the disease that people get. It's the sickness, right? With the signs and symptoms. This up here, SARS-CoV-2, is the actual virus. So this is a severe acute respiratory syndrome, coronavirus 2. It's a heck of a name. Scientists can't give anything names that are easy. The reason there's a 2 there is because, as we're going to talk about in a minute, this is not the first of these viruses. SARS-CoV-1 popped up in the early 2000s, and we'll talk a little bit about that in just a minute. So building on that, the first thing I want to do is talk about all those bugs out there and the ways that they can infect us and how they're similar and different to each other. The reason we're talking about this before we get right into talking about coronaviruses is because these different types of organisms are everywhere. They're problems to us, but they have similarities and differences to each other and to us that have real implications about how we treat them and how pathogenic they can be. So first, this is just a kind of bacteria called Staphylococcus aureus. It's just up there so I can talk about bacteria in general. So the first thing about bacteria we should talk about that's different from viruses is that bacteria are actually alive, right? That means they, in many cases, can move around their environment. They can take in sugars and use those to make energy. They can reproduce. Inside of those bacteria are many chemical reactions going on that are necessary to life. So why are we making such a big deal about bacteria being alive? And the answer to that is that viruses are not. Viruses are not living organisms. In nerd speak, they're obligate intracellular parasites. If a virus is sitting out here on a bench top or outside of a cell, it is completely inert. It can't do anything. It can't bring in sugar. It can't reproduce on its own. It can't move on its own. A virus can literally do nothing unless it's inside one of our cells. So because of that, right off the bat, comparing these two, there are huge differences, right? So we know that for a lot of bacteria, we can treat those with antibiotics. Probably most people here have taken the antibiotic penicillin. Penicillin works pretty well even these days against some types of bacteria. So bacteria are alive, right? They do all the things we associate with being alive. We can treat them with antibiotics. Antibiotic really technically means anti-life. And right off the bat, there's another difference between bacteria and viruses. Viruses are not alive, therefore antibiotics will not work on them. If you have a virus infection and you take a bunch of penicillin, you can take as much as you want for as long as you want, and it's not going to do anything for you. The last thing about bacteria is that they are relatively small, very small compared to our cells, but they're huge compared to viruses. And we'll talk more about that in a second. So those are the bacteria, right? So over here, this is just an example of a fungi. This is Coccidioides. I picked this because it's relevant to us in Arizona because it's what causes valley fever, which is really prevalent in the Southwest, especially Phoenix, right? Dust, people inhale the dust from construction and all that, and that will allow the fungal organism to take hold, and it can cause all kinds of problems. It can be fatal. Now, we have drugs to treat fungal infections, like Coccidioides. But a lot of times, most of the time, those drugs have severe side effects compared to the antibiotics we take for bacteria. And so the question is, why is that, right? And this goes back to us comparing these organisms so we can realize the uniqueness of viruses, right? So these fungal cells here are actually more similar to our cells than they are to bacteria, which is pretty crazy because they look a lot more like bacteria. Bacteria are what are called prokaryotic organisms. Fungi are what are called eukaryotic organisms. We as humans are also eukaryotic organisms. So our cells are more similar to this fungal cell here than the fungal cell is to the bacteria. What that means for practical matters is when we want to take drugs internally that are going to be able to kill those fungal cells, they're quite likely going to be extremely toxic to our cells as well, because our cells are so similar to fungal cells. So these both look kind of similar, microscopic organisms, but they are fundamentally different in a biochemical way. And over here, these are the parasites. I just picked Toxocara because it's the most common one in the US. So this people can get from their cats and their dogs. It's relatively rare in the US, but it can cause problems with your eyes and liver and all of that. These organisms are also much more similar to us than they are bacteria. So the drugs to treat these types of infections tend to have some pretty significant side effects as well. So those are three different types of organisms that can infect us and cause all kinds of problems. And it's different how we treat them because of how similar they are to each other and to us as compared to bacteria. But these three all have one thing in common. They're alive, right? So now we get to the viruses, by far the most interesting class of intracellular obligate parasites that there ever was. I'm really fascinated by viruses. There's many, many different kinds that do things very, very differently, but they're extremely successful organisms. They are incredibly tiny, as we'll talk about in a second. And yet they are, if you look at a gene level or DNA level, the most common organism on this earth because there's just so many of them. And they're all extremely varied in how they do things and the different kinds of cells that they can infect, but they're extremely successful. So we're going to focus on the viruses because we're talking about coronaviruses. Now, a couple of things about viruses in general before we move on. One is they are tiny, right? It's kind of hard to talk about how small microscopic things are compared to each other. So I like to blow things way up, right? So if you're talking about one of our cells, so to build a human being, you need about tens of trillions of cells, give or take, depending on your size, and you need 200 different cell types. Right? And those cells are different sizes, but roughly if you took one human cell and made it as big as an entire professional baseball park, like I'm not sure if it's Bank One ballpark down in Phoenix still or not, but you take a professional ballpark, the entire thing, the field, the stands, the concessions, everything. If one of our cells was that big, then a bacteria would be about as big as the infield, maybe a little bit bigger than the pitcher's mound, right? And a lot of viruses would be significantly smaller than the baseball, right? So they are just incredibly tiny and not alive. So antibiotics don't work on them, right? We do have drugs that are antivirals, but they're not truly antibiotics. So one of our main defenses that we use to combat viral infection is vaccines, if we can make a vaccine for the virus, right? We know there's clearly vaccines for some viruses like polio, right? But there's not vaccines for some viruses like HIV. And we can talk about why that is at the end if people want to. So for the rest of this, we're going to focus on a specific virus, right? So we're going to talk about coronaviruses. So we know there's about 7 different types of coronaviruses. They were discovered in probably the mid-1960s. And 4 of those just cause the symptoms that we associate with, a bad cold or maybe a little touch of the flu. They're not really that pathogenic, but there's some exceptions, and those exceptions are shown here, right? So SARS-CoV-1 first popped up in the early 2000s, 2002 in southern China. And this virus, as you can see the numbers there, it infected about 8,000 people, killed roughly 700 people, and had almost a 10% fatality rate, right? So this virus actually, it popped up, right? It appeared and people got sick from it. And the second round of infection was mostly healthcare workers trying to help them, but not knowing what the virus was because it was brand new, right? It had just emerged. And so those people fell ill as well. That was very quickly kind of cordoned off, right? And so that virus didn't get a chance to spread beyond southern China, really. And so because of that, that outbreak was brought under control with relatively small numbers of infections and death, but it still had a pretty high fatality rate. And so SARS-CoV-1 disappeared, right, as viruses do. And so about 10 years later is when another coronavirus appeared. This is Middle Eastern Respiratory Syndrome, or MERS. So this appeared in Saudi Arabia in 2012. This is thought to be spread from camels. There's some good evidence showing that. Here, we only had, I say only relatively, we had about 2,500 people infected, but it killed 882 of them. So that's a 34.4% fatality rate. That kind of sent shivers around the necks of people who work in infectious disease around the world, because that is an incredibly high fatality rate. Luckily, that virus didn't spread very well from person to person. Had it mutated so it became effective at spreading well from person to person, it could be a whole different world today. But the fact is, it didn't. And it was quickly tamped down by isolating people very, very quickly. And so MERS also went away. So to put these numbers in context, right, 9.6%, 34.4%, we can talk about the numbers for probably the pandemic that everyone knows about, right? And that's the Spanish flu of 1918, 1919. Back then, the world population was about 2 billion, right? And they think about 25% of the world was actually infected with the virus, and about 50 million people actually died from it. So if you do all that math, it's about a 10% fatality rate. So about the same as SARS-CoV-1. And we know what a catastrophe that was. So that's one reason. I mean, it seems like a long time ago now, right? But it really wasn't. When SARS-CoV-2 first showed up, these are the fatality rates that we had to compare it to. And there for weeks, it wasn't really known what that fatality rate was going to be. Luckily, it was significantly lower than that. But in some ways, I feel like we were really lucky to have that happen. So to update us where we are now today with SARS-CoV-2, this started in Wuhan, China. I think probably everyone knows that. These numbers are specific to the US after this, unlike these up here. So right now in the US, we've had about 32.6 million people infected and about 580,000 people have died. And that translates into roughly a 1.8% fatality rate. So it's actually interesting. So here's the numbers on the 3 very pathogenic coronaviruses that have shown up in the last 21 years. Some data just came out yesterday from some research that's being done at the University of Washington, and they're firmly convinced that the world has really underestimated the amount of COVID deaths. So if you're interested in reading about their research and some of the conclusions that they've come to, you can Google this link right here. And by the way, this link is a little misleading. Since you can't have periods in this address, that's actually 6.9 million, not 69. They're not saying 69 million people have died from COVID. They're saying 6.9 million worldwide have. So what they do is go through and in this article, they present how they come up with their conclusions that basically probably about 950,000 people in the US have died from COVID. And throughout the world, different countries have underestimated by different amounts. Their hypothesis is that Russia has vastly underestimated by about 5-fold the number of people that have died from COVID. So this is pretty not controversial at all, right? This is kind of history. This has been known for quite a while. This is where we think we are in the US right now, as the years pass, and this is, is in the rearview mirror and we can really examine it in detail. These numbers will probably become, a lot more accurate and may change a little bit, maybe up like this article would suggest, or maybe down. But if you want to read more about this, then just Google this article. And we can send you this if you want to. I'm going to do this a lot in this presentation, by the way, talk about a few things. And then if you're really interested in it, I'm going to direct you to a place where you can learn more about it. So that's kind of where we are as of today, right? I got these numbers. They were accurate yesterday. So to talk about how this whole process is going to work, now that we've had an introduction to coronaviruses, what I want to talk about next is just a little human cell biology. So everything on this slide has to do with an everyday normal human cell. There's nothing to do with the virus on this picture. Not yet anyway. So what we're looking at on this slide is how a normal cell functions, right? This is just a cell going about its ordinary life, not being hampered by virus infection, nothing like that. So just to kind of reiterate, in our bodies, we have tens of trillions of cells, right? We have about 200 different cell types. That's what it takes to build a human. And they have all kinds of different shapes and different functions, but we can symbolize them all with a picture like this. So this is going to be the plasma membrane that completely surrounds the cell, right? This is the cytoplasm in here. This is the nucleus, and inside of the nucleus is where all of our DNA is. So we got to talk about DNA a little bit so we can talk about mRNA, so we can talk about proteins. So to understand how these mRNA vaccines are going to function and how they're going to be built and why they work, we got to understand how cells just do things in an everyday manner. And we don't have to get into a lot of detail. We just need to talk about a couple different processes. So we're going to start inside here in the nucleus. Right? This is where our DNA is. So we know as humans, we have 46 chromosomes, right? We have 46 chromosomes, which is just our entire library of DNA. Those 46 pieces of DNA have about 3.2 billion base pairs of DNA to make all of that up. And in all that information, there's roughly about 22,000 genes. And each one of those genes has a set of instructions on how the cell should build one single protein. So that's what a gene is. It's a little section of DNA, right? It's not billions of DNA nucleotides long. It's maybe a few thousand, right? But one gene has a set of instructions on how to build one protein. So let's talk about why you would want to do that. And to do that, I want to talk about a specific protein because I think it's one that most people are familiar with. So most people know about insulin, right? And how insulin works. Insulin is a hormone, but it's also a protein. So that's why we're talking about insulin right now, because it's our example of a protein. So backing away from this slide, big picture point of view, when I get done with this presentation, if I go eat a bunch of sugar, the level of sugar in my blood's going to go up. It can't go up too high because that'd be a bad thing. Right? It causes all kinds of problems. So my pancreas is going to release the insulin hormone or insulin protein. It's going to go around to my cells and tell them to take that sugar out of the bloodstream, bring it inside. That's going to bring my blood sugar back down to normal. And it's also going to make that sugar available to the cells to make energy with, right? That's the whole point. So later on, I may not eat any sugar at all tomorrow. So I may not need any insulin released tomorrow at all, but maybe the day after I eat a ton of sugar. So I need a lot of insulin to be made. So the point is, as the days pass in someone's life, sometimes they're going to need a lot of insulin. Sometimes they're not going to need any. Sometimes they're going to need a little bit, right? So we need to have a way to make sure we're always making the right amount of insulin protein in our body. So to do that, if you think about it, one thing we're going to have is a place to store the instructions on how you build an insulin protein. And this set of instructions can't be damaged when I use it. It has to be like blueprints for a house. I look at it, okay, that's how I build that. Then I put my blueprints away, but I can come back to my blueprints the next week and be like, okay, I need to look at that again. That's how I build that, and then leave them alone. So a gene is the same way. It's a set of instructions, like a set of blueprints, but we can't change it when we use it because we may need to look at it over and over and over because I may need to make insulin every day. So because of that, if you go in the nucleus of our cells where the DNA is, we have a gene for insulin. On one of those chromosomes, there's a short section of DNA that has the exact instructions on how we make the insulin protein. I can use that today, and when I'm done, I'm going to put it back, so to speak, and it's going to be completely unchanged, completely unmodified. And if I need it again tomorrow to use as a set of instructions, it's going to be there. So that's the importance of the DNA. It has the genes, and the genes are the sets of instructions on how to build all the proteins that I need. So let's say I need some insulin because I just ate a bunch of sugar. What I'm going to do is go to my DNA, find the right chromosome, find the area of the chromosome that has that gene for insulin, and then using a process called transcription, I'm going to make a messenger RNA copy of that insulin gene. And when I'm done with that, I'm going to do two things. I'm going to take my messenger RNA copy of the insulin gene, and I'm going to bring it out in the nucleus. We'll come back to it in a second. The next thing I'm going to do is take my DNA and essentially put it back on the shelf. It's going to be completely unchanged by this whole process. It's there waiting for me tomorrow if I need to make more insulin. So the story of DNA is done for this slide, right? But we have a messenger RNA copy of that insulin gene. So we're going to bring that out into the cytoplasm of the cell. Now, this is the exact same thing. It's just drawn bigger. So this messenger RNA for our insulin is the same as this one. We just want to show more detail on it. So when a messenger RNA gets out into the cytoplasm of the cell, and only when it gets out into the cytoplasm of the cell, being in the nucleus will do it no good as far as being translated into a protein. The mRNA or messenger RNA has to go into the cytoplasm. And once it gets out there, an organelle called a ribosome is going to find it, bind to it, and then, long story short, with the help of these little structures here called tRNAs, the ribosomes are going to take this messenger RNA, read its set of instructions, and use that set of instructions to build an insulin protein, right? So we know proteins are made out of amino acids. Like, we eat those. When we get protein, our body brings them inside and uses them to assemble our own proteins. So at the end of this, we have our functional insulin protein, which we can kick out of the cell, kick out of the pancreas, and then it can go do its job. So that's the gist of it. The genes are in the DNA. A gene is a set of instructions on how to build one protein. When it's time to build that protein, we transcribe that one gene into messenger RNA. Then we take that messenger RNA out into the cytoplasm, and using a ribosome, we translate it into protein. Now we have our functional protein. Now we have our insulin in our example. Now, if we take a look at what happens next, this DNA is completely untouched, completely unchanged, and is going to live for the life of the cell. It's extremely stable. It could live for years. Messenger RNA is only going to live for probably minutes to hours at most. Messenger RNA is super unstable, so we're going to use it as a set of instructions to build a protein, then it's going to get broken down, right? That's going to be important for how we make our vaccine here in just a minute. So this is just gene expression. This is how we make a protein. But it's important to understand the general steps so we can talk about how how we build this vaccine and the process by which it works. So when we talked about this slide here, we talked about how, in general terms, we build a protein. Now, we make tens of thousands of proteins, and some of those proteins, not anywhere near all of them, but some of them are inserted in the plasma membrane of our cell. So my little stop signs and triangles here, I don't know what you call that. These are meant to represent proteins that are sticking outside the cell. They're embedded in the plasma membrane, so they're part of the cell, but they're sticking out into the extracellular fluid. Right? So that happens for a whole bunch of reasons we don't really need to get into. What's important about these proteins right now is that our immune system knows every single one of our extracellular proteins, right? So if immune system cell comes up to the cell you're looking at right here, it's going to take a look at this molecule, this protein. It's going to look at this protein. It's going to look at this protein, and it's going to say all of those belong to me. These are all self proteins. This cell is healthy. This cell is not infected with a virus. This isn't a virus. This isn't a bacteria. This is a normal, healthy cell. So one purpose out of a bunch that these proteins on the surface of our cells have is they identify us to our immune system cells. Immune system cells aren't allowed to be active until they learn every single molecule that could be on the surface of one of our cells. And so if this is the whole picture, and all our immune system cells see are the self molecules on the surface that should be there, then our immune system will never do anything to this cell, right? So there's like 8 weeks of cell biology condensed down into like 5 minutes, right? So we're going to come back to this picture, and we're going to build on this and talk about how we actually make our vaccine, what it does when it gets inside of this cell, and then what our immune system's response is. But this is the picture we're going to build on. But what we want to do is leave this cell for a while, leave this uninfected, completely normal human cell, and we're going to talk about the coronavirus. So if you look at this, it's roughly the same size as that last slide. But keep in mind, in real life, this is minuscule to that slide on the last slide, right? You could fit tens of thousands of coronaviruses inside of one single human cell. So to talk about coronaviruses in a little bit of detail, we can look at this picture, right? So a few things. So first, as humans, we talked about how we make proteins on the last slide, and we can make literally tens of thousands of proteins. Coronaviruses make about 37, right? 37 compared to tens of thousands. Viruses are tiny, right? Our genome is 46 separate pieces of DNA, and it's 3.2 billion base pairs. The genome of a coronavirus is actually RNA, but the important part is it's about 30,000 nucleotides long. 30,000, not 3.2 billion, right? So it's tiny compared to our cells. So it makes about 37 proteins, or you need about 37 proteins to build a coronavirus, but there's just a couple major ones shown here. And the one we really care about is the spike protein. So the spike protein sticks out the outside of the virus. And this looks kind of like a crown. That's why they're called coronaviruses, right? So these other proteins, right, and there's many more. There's about 35 more, 34 more, something like that. Their roles really aren't important for this discussion. We're going to focus entirely on this spike protein because it's sticking out of the virus, sticking out into the extracellular space. And as we're going to see in a second, when a coronavirus contacts one of our cells, it's going to use this spike protein as a key to get into our cells. So we're going to come back to that spike protein in a second. So inside here, this is the genome of the coronavirus, right? So it has all the information it needs to force our cells to make about 37 different proteins, right? So there's information in here to make 37 different proteins. That's important because we're going to come back here and talk about how what we're going to do eventually is cut out a little tiny chunk of this virus's genome, just the section that has the instructions on how to make the spike protein. That literally is going to be what our vaccine's made of, but we're going to get back to that in just a minute. First thing I want to do is talk about the structure of this tiny little coronavirus. So this was our cell, right? This is what the virus looks like. So the next question is, what happens when a coronavirus actually infects one of our cells? How does that happen? And that's what this picture is showing. Now, to be honest, I stole all these pictures off the internet. So this whole picture of a cell right here is exactly the same as this one, right? And this picture of this coronavirus right here, that's what this is. It's just different pictures. So this is a coronavirus, right? Infecting a completely normal human cell. Only now the infection is going to actually happen. So what happens is this cell is just undergoing its ordinary life, minding its own business, and then this coronavirus is going to come along. And that coronavirus is going to use its spike protein, right? So the spike protein there is the exact same thing as the ones here. So those spike proteins are going to be the key that get the coronavirus into the cell. We know how keys work, right? So keys work by putting them into a lock and then turning them, and that opens the lock up. This is really not that much different. So we need a lock, right? So the lock is going to be these receptors on the surface of our cells. So these are just normal old surface proteins on our cells that have functions that we need. For example, one of the receptors that the spike protein of coronavirus can bind to is called ACE2. This is angiotensin converting enzyme. This is involved in regulating our blood pressure if we're dehydrated or we get a really bad cut or something. This is something that we use as humans for our own biochemical pathways, but viruses are going to hijack those. So for us, we use this to regulate our blood pressure, but for the coronavirus, this is going to be the lock for the spike key, right? So that has an implication though. A coronavirus can only infect a cell if that cell has this ACE2 receptor on it, or a different one called DPP4. Doesn't really matter what that is. They're just receptor proteins. They all have normal functions in our cells, but the virus is going to hijack them. So if a cell doesn't have ACE2 or DPP4 or a receptor that the spike protein can bind to, the virus can never get in. It'll just bump into the cell like hitting a brick wall and nothing will ever happen, right? But that spike protein is a key that binds to this lock, which is the receptor on the surface of our cells. And if that binding happens, it's going to trick our cell's machinery to bring in that virus inside. Now this cell's in big trouble, right? The virus has attached, it's penetrated, as it's called. And then the next thing that's going to happen is this virus is going to completely uncoat and expose its genome. So this is the virus's genome. Now it's free in the cell. Now it's the exact same thing as this, right? So all of this other stuff, all those 35 other proteins have all melted away, uncoated. And so now this is viral RNA. But here's the thing, this viral RNA looks like our RNA. And the virus can set it up in such a way that it tricks our cell into treating the virus's RNA like the cell's own RNA, because the cell can't tell the difference. So all that machinery we talked about here that takes a messenger RNA and a ribosome binds to it and builds proteins, all of this happens in this infected cell because the cell can't tell that this isn't human RNA, right? Once it gets to the messenger RNA form, the host cell is going to translate into protein, unwittingly making virus proteins for the coronavirus, right? So that's what happens next. We're going to make some virus proteins by the hundreds. And then we're going to make a whole bunch of virus genomes by the hundreds. And then we're going to assemble them together in brand new coronaviruses. And then hundreds of brand new infectious coronaviruses are going to bud out of the cell, killing it in the process. And now we have hundreds of brand new coronaviruses that can then go on and do this process to brand new uninfected cells. This is why virus infections can get wildly out of control, because one cell comes in, hundreds leave. And those hundreds disperse to go find new uninfected cells. And can you see how fast the numbers of virus infected cells can grow? Because every time one goes in, potentially hundreds can go out and find new uninfected cells to infect. So this is what the virus wants to do, right? And so all the signs and symptoms associated with different viral infections happen because they are forcing our cells to make new viruses because they're not alive and they can't do it. And in the process, it's killing our cells. So fundamentally, at the end of it all the different viruses, HIV, influenza, hepatitis, the list goes on and on and on. They cause the problems that they do because of the cells they infect, and then they kill those cells. And then whatever function those cells had, we don't have those cells anymore. So whatever the job that cell was performing is not being done anymore. And that causes all kinds of signs and symptoms and diseases depending on the virus. So the real key here for this figure is this interaction right here. If the spike protein of the coronavirus can bind to a cell surface receptor like ACE2 on our cells, this is the key going into the lock that gets the virus in. So if you think about it, if we can interrupt this reaction, if we can stop the virus, even if it's in our body, from binding to the receptor, we're never going to have a problem because these viruses aren't alive. They can't replicate or make new copies of themselves unless they can get inside of a cell, right? So if we could prevent this interaction from happening, then maybe we were infected with a few hundred coronavirus because someone coughed in our face, but they'll bounce around out here in the extracellular fluid, eventually get broken down, and nothing will ever happen. We probably won't even know they were ever in our body, right? So one key to what we're going to talk about here coming up is we really want to stop this from happening. If we can keep that virus from getting in, it can't replicate. It can't make more copies of itself. So, quite a while ago now, over a year, almost a year and a half, I guess, the SARS-CoV-2 appeared in the world, started infecting people, started doing this process right here. So now we have a problem, right? We need to stop this worldwide pandemic as soon as we possibly can. And so the way that we chose to do that was by making a vaccine. And so we made a vaccine in record time, I think some people would say. But what I'm going to argue and show you why I feel this way is we've been making this vaccine for a very long time. We've been making this vaccine since 1990. We just didn't know it, right? And here's why. So if we're going to prevent this virus infection from happening, if we're going to make it so people are vaccinated for SARS-CoV-2, we need to make a vaccine. So the first step of making a vaccine is knowing what part of the virus that you should make the vaccine to. There's 37 different proteins. How do you know which one? You want to find a part of the virus that's a good target for the immune system. So that's where we start this story. So we know there's SARS-CoV-1, right? That came about, like we talked about, 2002. And after SARS-CoV-1 disappeared, a lot of research was done, basic research was done on SARS-CoV-1 because it had the capacity to be catastrophic, right? Like SARS-CoV-2 is now, only its mortality rate is 5 times higher. So people started to look and started to try to build and find ways to make a vaccine to a coronavirus like that. So this is some more light reading if you want. So this is a research paper that came out in 2006. And the gist of this is people took the SARS-CoV-1 and did a huge array of tests to find out exactly what the part of the virus would be best to make a vaccine to. And their conclusion in 2006 was that it was the spike protein. So when SARS-CoV-2 came out a little over a year ago, those spike proteins are very similar. So it's not hard to make that leap that if we're going to make a vaccine, we know from all this previous research that the SARS-CoV-1 spike protein looked like it would work really, really well. So Moderna says, let's make a vaccine for the SARS-CoV-2 spike protein, because based on this research, it's quite likely, you never know till you test it, but it's quite likely to be extremely immunogenic and work well as a vaccine. So reason number one, we built this vaccine so fast is we already knew what to make it to. It had to be tested, of course, but luckily enough, this work done here totally panned out for SARS-CoV-2. So that's step number one. We know what to make the vaccine to, what part of the virus to target our immune system toward, right? Now, how do we build the vaccine? So that's where we come to how you make an mRNA vaccine. So I'm going to go over like the kind of the major steps of this, but if you want to read more about messenger RNA being used for vaccines and other things, this is a really good readable paper. Right, so this is mRNA Vaccines, a New Era in Vaccinology. This came out in 2018. So this is reason number two that we made the vaccine so fast. The groundwork had been worked on for this since 1990 because the power of this is amazing. It's going, here's, you know, this is me talking, not talking about data, talking my opinion just for a second. You're going to start to see mRNA vaccines being used for almost everything. You're also going to see a lot of anti-cancer drugs delivered using this kind of system because it's cheap, it's safe, and it's fast, right? Those three things are pretty unbeatable. And also, we're not going to go too far down this pathway, but a lot of times the delivery system that you have to deliver a vaccine or a drug can itself, you know, be immunogenic and excite our immune system. So you can't use the same system over and over. But with this technology, you can. It's not immunogenic in that way. So people started to figure out how to do this process that I'm going to talk about in like the early 1990s. But it really didn't, you know, start to really work in a productive, you know, way that was easily scaled up. Right? It's one thing to be able to do this super intricate kind of experiment or construction in a laboratory, you know, where I use $5,000 worth of reagents and it takes me 2 weeks to do. That's not helpful for making a vaccine for billions of people. So part of this process was making this process work. And then once it worked, making it scalable. And now we can do all of those things. Like in my opinion, we'll see if it pans out, making an opinion on something that's being recorded. I think flu vaccines are going to start to be made this way. It's much smarter because it's so much faster. It takes too long to make a flu vaccine. That's why they're only about 75% effective, something like that. We can respond to viral evolution faster now because if they evolve and change the shape of their outer proteins, like their HA and their NA, we can make vaccines to those much, much quicker. So it should make flu vaccines work much better because we can make them a lot faster in response to changes that the virus may do. Anyway, back to this story. So the reason I have this here is to review the whole structure of SARS-CoV-2, right? Remembering from the last slide, what we really want to do is introduce our immune system to this spike protein, enable our immune system to fight this spike protein because we're pretty sure that'll work from this work done here in 2006, right? So here's what we do. This is why these vaccines are so safe, one of the reasons. What we're going to do is we're going to throw away all the spike proteins, all the other 36 proteins that make up this virus. We're going to take the whole genome, which has the information to, you know, to express all 37 of those proteins. We're going to find that one little tiny section that has the instructions on how to just build the spike protein. We're going to snip that out, and there it is, right? So all the rest of this gets thrown away. The only thing in that vaccine is a tiny chunk of the genome, no proteins, None of the instructions to make the other 36 proteins, just that section of the genome, the mRNA that has the instructions to just build the spike protein. That's it, right? But now this is good because if you think about it, what we can do is take this messenger RNA and you can put it inside of our cells. And if we make this messenger RNA look like our messenger RNA, then our cells will make the spike protein, which is going to be how we're going to make the vaccine, as we'll see here in a second. But the only thing our cells are going to make is this spike protein. The other 36 proteins aren't there. There's no virus there at all. It's just that one single protein. But here's why this took so long. As trivial as this seems, it's really not. This, if we inject this into someone's arm, it's never going to make it inside the cells, right? And the reason for that is the plasma membrane of our cells is what's called lipid soluble, right? So some things that are lipid soluble, which means they can melt right through it, like some of the sex hormones and some of the vitamins like D, E, A, and K are all fat soluble vitamins. Those types of molecules can melt through our plasma membrane and get inside of our cells. There's not very many compounds like that. Most compounds are what we call water soluble. When water soluble compounds hit the plasma membrane of our cells, like hitting a brick wall, there's no way they're getting in. So mRNA is water soluble. So if I were to take this, put it into a syringe and shoot into someone's arm, it would come up to one of their cells and bounce off and bounce off. It'd never get inside, right? And mRNA is super unstable, like we talked about before. It might last for a few minutes, maybe a few hours at most. So I would inject that into somebody, it would never get into their cells, and then it would be broken down, and that would do me absolutely no good, right? But what Moderna and Pfizer have learned how to do from the basic research, this is a great story. And it's not like there's a lot of science in there, but it's pretty accessible to pretty much anybody if you want to read about how this happened, like the last 30 years or so. So what we're going to do here is take this messenger RNA and we're going to make it lipid soluble, right? And so what we're going to do is we're going to surround it by these little compounds. We're going to emulsify it, which is just nerd speak for coating, right? We're going to coat this thing, coat the messenger RNA with these compounds that turn this water-soluble messenger RNA into a lipid-soluble particle. So this literally is what's going to be injected into your arm if you're taking the Pfizer or the Moderna vaccine. So if we go to that, this probably looks familiar, right? So here's our normal old cell. So this is a cell from the person that just went and got in line to get their vaccine, right? So they get the shot, someone injects them with it. This is what happens. This messenger RNA is able to get through their plasma membrane because it's lipid soluble. Right? So this messenger RNA is now inside the cytoplasm of one of our cells, right? So in real life, you have a syringe, right? So you injected, probably a few hundred, few thousand of these molecules. So this process we're looking at here is going to happen to a few hundred, maybe low thousands of our cells. These cells are going to end up getting killed, but that's okay. We'll just replace them. But in the meantime, we're going to have a tremendous benefit, as we'll talk about here in a second. But what has happened now after you got that shot is this messenger RNA that has the instructions on how to build spike proteins is now inside your cells. That's it. None of the rest of this is ever anywhere near your cells. But once we have this mRNA inside of our cells, this coating is going to come off. And now we have messenger RNA for the spike protein inside of our cells. Now, I told you I stole these pictures online. So this messenger RNA for our insulin protein is exactly like this messenger RNA for the virus spike protein. Everything that the ribosome would use to recognize it is, hey, that's one of my messenger RNAs. I have to translate it, make a protein. Ours looks like this, right? So the whole process that happened here, we're going to fool our cells' ribosomes and some other parts of it, and they are going to take this virus spike protein and translate it into protein. And once that happens, a few hundred to a few thousand of our cells are going to make the virus spike proteins and stick them on their surface. So now we have this human cell with virus spike proteins sticking out of the cell. Now, before, when we talked about this picture, we talked about how our immune system cells would come up to this cell, look at all these proteins, and know that they were self, and then leave the cell alone, go away. That's going to change now, right? And the reason that's going to change is because the immune system cells are going to see the spike protein. And they're going to say, that is not one of my proteins, that is not self. This is something foreign and we should kill it, right? And that's exactly what's going to happen. Your body's going to mount an immune response to some of your cells, a few hundred or a few thousand, because they are expressing a virus protein. And the immune system cells know that's not us. That's not what's called self in immunology. So the next step is the immune system is going to find those spike proteins and wipe this cell out, right? But in doing so, it's going to do something pretty wondrous for us. And so that's what we're going to talk about next. So now we've lost our cells, but our immune system has also learned about this new protein that is non-self. And it's going to learn how to kill it. And more importantly, it's going to remember for a very long time how to kill it quickly. So here's what we're going to get, right? So we lost all those cells, but here's a couple of things we're going to get. We're going to have some cells called B cells that are going to become plasma cells. They're going to make antibodies to that spike protein. And we'll talk about what that does here in just a second. Probably even more importantly, we're going to get two more things. We're going to get what are called memory cells. We're going to get memory B cells and memory T cells. So think about it this way. The first time you get infected with an organism, whether it's real or whether it's a vaccine, which isn't an organism, but it's something new in your body, you have a tiny handful of immune system cells that are capable of recognizing it and attacking it. So if that process happens, naturally, you're probably going to get pretty sick and then you're going to get better. But what you have after that is massive numbers of memory cells. So if that organism ever shows up again, instead of having a handful of cells that attack it, you're going to have thousands, right? And vaccination does the same thing. If you teach the body how to fight the spike protein by using that process we just talked about, then you're going to have memory cells by the thousands that also know how to fight that spike protein. So that's going to prime you for a future infection, as we're going to see here in a second. There's no way to get these unless you get SARS-CoV-2 infected for real or you get vaccinated. That's the only way to build these memory cells. So here's our virus, right? So you were vaccinated, and then all of a sudden, you know, a month later, you were actually exposed to SARS-CoV-2, right? So what's the next step here? What's going to happen? And the answer to that is those antibodies are lying in wait, right? And when those antibodies are in your bloodstream, and so to kind of back up for a second, this is an amazing thing. In your lymph nodes, right? That's where B cells become plasma cells. And plasma cells live in your lymph nodes. And every single day, a plasma cell makes about 1 billion antibodies. So think about that. Thousands and thousands of plasma cells, each one of them making 1 billion antibodies every day. Those are spilling out of your lymph nodes, entering your lymphatic system, which is connected to your cardiovascular system. So your body is just awash for a couple weeks in these antibodies. So what's going to happen is these antibodies are going to find the spike protein and they're going to bind to it. That's going to do a couple things, right? Because remember, I told you that it was so important if you were the virus, you had to be able to use that spike protein as your key to bind to this receptor, which is the lock. So if this key or the spike protein is covered with antibodies, it's like taking your key and wrapping it up in duct tape or something. There's no way it's going into the lock. So one thing those antibodies binding to the spike proteins does, it prevents them from ever getting inside the cells because the spike protein can't bind to the receptor. We took the connection between the key and the lock and we interrupted it. Just literally, I mean, it's a simplistic example, but it's 100% accurate. It's basically like wrapping a key up in duct tape, only we're wrapping the spike protein up in antibodies. It's going to do the same thing. Now, there's also a whole host of other things happening. There's immune system cells that recognize this complex of an antibody bound to something, and they engulf the entire thing and destroy it by a process called phagocytosis. There's a whole other kind of cell, a family of cells called T cells that are involved in this, but this is kind of the gist of it. We taught our body how to fight that spike protein. So if that virus ever had the audacity to show up, we already know how to kill it. Right? And there's one more component to this story that is really important. So the virus is gone. And the other component is this, right? I'm not sure why anyone would ever show anyone a graph on a presentation on a Friday evening, but bear with me for just a second. This slide really shows you the power of vaccination, because here's what we're looking at. If you look at this graph here on the y-axis, this is blood antibody level. So the higher we go, the more antibodies we have. So the higher we go up this line, the more of these guys we have, right? This here is the passage of time. And here we're talking about days. So before we talk about vaccination, let's talk about what happens in a natural infection. So let's say you get infected with a particular organism. That's day 0. You get infected, let's say it's flu. Let's say you get infected with a flu virus at day 0. So a couple of days are going to pass, right? You're going to start to make an antibody called IgM. And then after about a week, you're going to start to make a little bit of this antibody called IgG. And about 12 days after you were exposed and that organism started to multiply inside of your body, you're making the most IgG antibody you're ever going to have. This doesn't really matter what these are. They're just antibodies that our body's using to fight the virus. The point is, after 12 days, you have high levels of IgG antibodies. So that means for 12 days, your body's been fighting off that virus. Hopefully it's winning, but sometimes human bodies lose because it takes a long time at first exposure or the first time you're infected with an organism to make this many antibodies, right? Now, let's say you survive this infection. So then the levels of antibodies go down, but notice they're not completely gone. And then this time period here is 2 weeks roughly. But in real life, this could be weeks, months, years, decades. So for pox virus vaccines, we know this time period could last for probably 40 years at least, right? So now what's going to happen on this graph 2 weeks later, but in real life, maybe years later, the same exact organism infects this person, right? But now what's going to happen is called your secondary immune response. And so when your secondary immune response, look what happened. When your secondary immune response happens, look how different it is. It's not going to take, a week to start to make IgG. It's going to happen in hours, right? And it's not going to take, if you look here after 12 days, we have this much IgG. Look how much we're going to get this time, right? That's why once you survive a natural infection, if you're ever infected with that exact same organism, again, it has to be the exact same one, you probably didn't even know you're infected because your secondary immune response is so much faster and so much more powerful. It makes so many more antibodies so much faster that you kill the organism so quick you probably didn't even know that it was there, right? So what you're doing, back to SARS-CoV-2 and back to the story of coronaviruses, when you get a vaccine, you're doing this, right? So you get your shot here and the mRNA for the spike protein goes into your cells make the spike protein, your immune system recognizes those cells that are making the viral protein and putting them on their surface. Your immune system cells kills those, makes memory cells and all of that, and then you're fine, right? But if you're already vaccinated and then you get exposed to SARS-CoV-2 for the first time, the real SARS-CoV-2, you're not going to have this happen. You're done with that. The vaccine did this for you. If you're exposed or infected with real SARS-CoV-2 after you're vaccinated, this is how you're going to respond. You're going to make huge levels of IgG much, much faster. And the data shows you're going to wipe that infection out pretty quickly, and you probably won't even know you're sick. You probably won't even know you were ever infected. So the difference between this primary and secondary immune response is massive, right? So we can get this over with naturally. Hopefully we'll survive it, right? And that's true for all organisms. But the beauty of vaccination is that if we can vaccinate someone in a safe manner, because they're never going to get sick, right? With a good, well-tested, well-designed vaccine, you're never going to get sick, but you can get your body through this stage, get your memory cells that we talked about a couple slides ago. And then if you're ever infected with the real organism, this is how you respond, not like this. So that's the beauty of vaccination. So that's all wonderful, right? But if you're going to shoot something into my body, it better be safe, right? So we're not just going to willy-nilly just start, that's a great idea. We can get this messenger RNA inside of these cells. That's awesome. Let's do it, right? That's not how things are going to happen. So once Moderna came up, and the other companies, I'm just using Moderna because it's a good example. Those vaccines still have to go through a number of specific phases that have certain goals, right? And so those are shown here. So this preclinical, this is what I was arguing started in 1990, trying to get messenger RNA stable and to last long enough to get it inside cells, to get it translated into protein, right? All that kind of stuff. is the preclinical part. It's the basic science of taking what you know about cell biology and immunology and virology and building something to take advantage of our own immune system, to teach it, right? So once we go through this phase, which can last for decades, right? What we're going to do then is go into phase 1 trials. And what we want to do is give this to a very small number of healthy people. We're not trying to see if it's efficacious or if it works. We want to see what happens when we put this into people. Like, there always could be something very, very unexpected. And the best way to control for that, at some point you have to try to put something into people, right? But you take a small number of healthy people and then give them a low dose. If everything's fine, give some people a higher dose. And then if everything's fine, give them a little higher dose. And then if that goes okay, then we go into phase 2. In phase 2, we know things are kind of probably maybe safe, right? So at this point, what we're going to do is actually give this to some people who are maybe suffering from the condition that we want to help. Or if we're giving a drug that can potentially benefit them, we need people that it could benefit. Like say it was a diabetes drug, right? Then we would have some diabetics in phase 2, not in phase 1. Phase 1, we just want healthy people just to see what's going to happen, make sure it's relatively safe. So in phase 2, what we're going to do is get some people, let's stick with our diabetic, and include some people who are diabetic, try different doses, see which doses work the best, so we can settle on a dose to use therapeutically to test, and then also make sure that it actually helps a small handful of people. Not enough to be like statistically, make it ironclad that this is an efficacious drug, but like, let's see if, if we give this to a relatively small number of people who are diabetic, does it help them? And is it safe? And what dose worked the best? So from there, if everything, proceeds as planned, then what we're going to do is take that dose and we're going to give it to a lot of people, right? So this is phase 3. So this is where we're going to give this drug to a whole big group of people. We're going to give a whole big group of people that's kind of the same makeup, a placebo, right? And then we're going to let a certain period of time pass and we're going to make sure it's safe and also see if it works. So a lot of times this phase 3 has about 1,000 to 3,000 total participants. For Moderna, that was about 30,000. I'll show you the actual data here in just a second. So if this goes well, right, then the FDA is going to go through all the data, look at everything. A committee with no conflict of interest is going to look at all the data, look at the results, and confirm that it's 2 things, safe and effective. And that's when they start giving it to people, right? And so that's not the end of it. So we know there's a lot of vaccinations out there now for SARS-CoV-2. In fact, I think I saw today a little over 100 million Americans are fully vaccinated, I think. And that's a third of our country almost, right? So it's a lot of people. And that brings us to phase 4. Right? So you continue to look, because maybe even though you had a large group of people in phase 3, maybe there's this really rare thing that's going to pop up every now and then. So you don't let your guard down and stop collecting data on the side effects of vaccinations just because it's in widespread deployment, right? You're still going to go into phase 4 and track that. So these are the phases that we're going to go through to test our vaccine and make sure it's safe. So if we go back to phase 3, this is a really important one, right? Does it work and is it safe? And so here's what Moderna did. So if you look at the top of my screen, this is one of those things, if you Google that, that's the exact title of the article in the New England Journal of Medicine that I stole all these figures from, talking about Moderna's actual results. Right. So I'm going to go through some of these slides relatively quickly, but they explain exactly how they came up with every term, how they defined every term, the parameters of every single thing you could think of. So if you really want to dig deep in the weeds on how they conducted this trial and, the different parameters of it, just Google that title. It'll come up and it's free. So here's what they did. Right. So we have about 30,000 people who are going to be on the trial. And they're going to divide them roughly into 2. So if you look down here, N is the number of people, right? So if we look in this very first shot, right, there's going to be 15,000 people here, and change getting the vaccine, and 15,000 and change getting a placebo. If you look over here at the second shot, it's going to be a little bit less. People always drop out of clinical trials, so those numbers are always going to be lower. But what they essentially did was divide 30,000 people into 2 groups. One group got 100 micrograms of the mRNA vaccine, mRNA 1273, right? The other people got a placebo. And they gave them day 1 was when they had the first shot, and then they waited 28 days and gave them the other, right? So one thing about this, all 30,000 people were 18 and older. That's why someone asked why, you know, it's only good for people who are 18 and older. And the reason is because that's the people, the age of the people who are in the clinical trial. That I bet most scientists would say, you know, if you gave that to a 14-year-old, it would be 100% completely fine. But the fact is you haven't tested that, right? So a lot of the rules that we're following right now for administering these vaccines, like the Moderna vaccine, are there because that was what was actually tested. So why do you need 2 shots? Would 1 shot work just as well? And my answer to that is maybe and likely, you know, but 2 is what was tested. And the testing started in October, I think, and kind of finished up in November. So eventually, my guess is you will see, you know, a phase 3 clinical trial done or some kind of trial done where you just give someone 1 shot of Moderna and see the efficacy of it then. But that hasn't been tested yet. Right. And so eventually, I think they already have, I'm not sure if the results are out. They'll give the vaccine to younger people, actually do the trial, get the results, and then change how they're administering that vaccine. So a lot of the rules come because that's how the trial was set up. You're not going to deviate that and start giving people the vaccine in an untested environment. If there weren't kids in the phase 3, there's not going to be kids when it's generally deployed. Right? You got to follow the same kind of format. So this is showing you some of the adverse events after the first dose and some adverse events after the second dose. And they're very definitely side effects, right? Pretty minimal and nothing really serious. So this data, if we want to look at it in more detail, we can look at here, right? So up here, and again, if you want to go through this in like a lot of detail, just Google that title. But these are all local events. Right, so let's talk about what these graphs are actually talking about. So this is at the site of injection. So this is going to be pain, right? This is redness, right? This is swelling at the site of injection, and this is swollen lymph nodes, right? So if you look here, there's 4 different columns. So it's placebo dose 1, placebo dose 2, which are definitely lower, right? And then there's the actual vaccine dose 1 and the actual vaccine dose 2, right? And so that's the same format for every one of these different variables. And you're sticking a needle into your arm, right? So there's definitely going to be some physical effects of that. Like vaccines very definitely have some pain and some inflammation and some other side effects that happen pretty much every time. So if we go down here, these are systemic events. So we have a fever, headache, fatigue, muscle pain, joint pain. That's what those words mean, nausea and vomiting or chills. And so you can see these same blocks of 4, placebos are always lower, right? And then the people actually getting the vaccine had much higher levels of all of these. And I think just anecdotally from talking to people, you know, around the college, I think probably most people would agree that their second shot had more side effects than the first one, right? So these are just general trends. So you can look at this and see how they track the different adverse events that could happen. But the way these are characterized, you know, in people in vaccinology, these are all really minor, right? Someone going into anaphylactic shock, that is a huge side effect, right? And that's entirely possible in a situation like this, right? There's people out there that if they ate peanuts, they could go into anaphylactic shock and die. Penicillin has saved more lives than maybe anything on earth. I'm personally allergic to penicillin. If I took it, it could quite likely kill me. I could quite likely go into anaphylactic shock. So those kinds of things definitely happen with foods, with medicines, with vaccines. So those are things to keep an eye out for. But thankfully, the cases of those have been incredibly small, incredibly minute. And I feel like now that we have 100 million people in the US alone fully vaccinated, that we're really hopefully building confidence that this truly really is a safe vaccine, even though it has what I think of as like a revolutionary kind of game-changing method for building the vaccine. Okay, so that's great. We have some side effects, right? They're pretty minor, relatively speaking, but more importantly, does this thing work? And so that's what the next couple slides show. So here, this is the cumulative COVID-19 incidences, right? This is the day since randomization. Randomization is when they take that group of 30,000 people and they randomize them, right? Now, it's interesting how they randomize them. And if you really want to get into the weeds with that, you should Google that title and get the paper. But it's not, it's random, but there's also things you have to take into account for. Like we know that, if you're below the age of 65 and you're obese, you are much, much, much more likely to have problems if you get infected with COVID. So these two groups are random, but you still want to have roughly the same amount of people who are under the age of 65 and obese. If that number was wildly skewed, then your data probably wouldn't make any sense. It wouldn't be as, you wouldn't have as much confidence in it. So it's randomized, but there's also different situations that are cast in a light to make sure that they're equal on both sides of the aisle. So whether you're getting the actual vaccine or a placebo. Something else would be like, if you had, 1,000 people who had asthma in that trial of 30,000 people, you wouldn't want all 1,000 of those in one group or the other. That could really skew your data. You would want to make sure that that pathology, which is associated with a pretty bad, end with COVID infection, you'd want to make sure that those were pretty evenly distributed. So it's randomized, but there's also variables that people make sure are randomized within that. So we're not going to go too much into the weeds with all these numbers, but here's the gist. The people that got the placebo, over time, that group got more and more and more COVID, right? Over here, this is the group that got the vaccine. It's definitely not 100%, right? Turns out it's like 94.1% effective, but there's some people it just did not work on, 5% of people. So that's pretty good for a vaccine, actually. So this just shows you how many people got COVID that had the placebo, and you see the number goes way up. And then you see here's the number of people, the incidence of COVID as in the group that was vaccinated. And then if we break that down into numbers, then it's going to look like this, right? So in that group of people, at the end, 14,550 got the vaccine, 14,598 got the placebo, symptomatic COVID 11, symptomatic COVID 185, severe COVID 0, and severe COVID 30. So they have pretty specific definitions for these, which are all in this paper if you want to read it. So if you take all of this together, the conclusion that the group came up with was that 2 doses of this mRNA vaccine are safe, 94% effective in persons 18 or older. And like I said, the reason they're saying 18 or older is because that's the age group that was included in the phase 3 clinical trial. There was no one younger than 18. So that's why that specific number is there. So now, this has been, I think, like 3 months now, roughly. 2.5, 3 months that we've been actually actively vaccinating people with the Moderna vaccine and the other ones. But so now we're in that last phase where we have millions and millions of people vaccinated. So we surveil and start to look for those really rare incidents of side effects that maybe we didn't catch in phase 3. And for the Moderna vaccine, to my knowledge, there's really none that have popped up. So that's a wonderful thing. So at the point of this, the end of this, this is how we're going to give the vaccine, and it's proven itself to be safe. So what I'd like to do now, talking a mile a minute for a while, is answer any questions you guys might have. But while I'm doing that, I want to go to this slide, because if you find this type of thing really interesting, and you need some summer reading or watching, these are some things you might look into. So if flu interests you, this is a really readable book, and it talks about the 1918 pandemic. And then something we didn't talk about, but was a question someone sent me from the audience was, could the 1918 flu pop up again? And it's a crazy story because this guy, Jeffrey Taubenberger, in the 90s, he went out to the permafrost in Alaska. In fact, so permafrost doesn't unfreeze, hence permafrost, right? So he found bodies of people who died of the 1918 flu and were buried in the permafrost, and they never thawed out. So if they died of the influenza virus, it could maybe be alive in them. And it turns out he found one, or he found more than one, actually. He found this woman who died of the 1918 flu, but she was really obese. And so the adipose tissue, when they buried her in the permafrost, protected the tissue with the virus in it. So when he dug her body up, as macabre as that is, in the 1990s, he actually isolated the 1918 flu virus out of it. So this tells you the story of all of that. And also, the whole story of the 1918 flu and even some of the politics, which is pretty awesome. This story is all about the Americas before Columbus showed up. And there's a whole bunch of, different topics in this book, but one of them has to do with immune systems and how different groups of people from different areas have slightly different immune systems. And so they respond differently to different viruses. And so one thing this talks about, so we talked about, the 1918 flu having a mortality rate of like 10%. We know now from looking at historical records that, there were millions of people in the Americas before Columbus showed up. And, when those people came from Europe, they brought diseases that had never been seen before, like smallpox and like influenza and all of that. So these viruses would sweep through communities and have mortality rates of 95% plus, which is just mind boggling to me. So this is another great read. This here is, we didn't really talk about this much. I intended to, but I always think I'm going to talk about way more than I do. This is talking about how advances in DNA sequencing and knowledge, which has been exponential in the last few years. It's now super cheap and super easy to sequence DNA, and we can now purify DNA from human skeletons that are 50,000 years old. And you put all those things together, they're doing really cool research to talk about how people spread throughout the globe. And then down here, this link is a Frontline show. It's 10 years old, but it is awesome. If you've ever heard about the whole, MMR vaccine causing autism and all that kind of thing, this tells you that whole story about the now discredited doctor who proposed that. Turns out he was being paid and all this kind of stuff. But, the whole, still in society today, there's still a large group of people that think vaccines can cause autism, which has been completely debunked in probably 25 countries at least. But that falsehood still sticks with us. So this show on PBS tells that whole story, interviews all the major players on both sides. And so now I think that you have a little bit of knowledge about cell biology and how vaccines are made. This is a fascinating watch. 10 years old and more about measles and autism, like nothing to do with COVID or anything, but it's a really good history of vaccines and what they've accomplished and some of the problems they've encountered in kind of regular society. So anyway, so that's my presentation. So it's Friday evening. So thank you very, very much for being here. And if you have any questions, I'd be more than happy to attempt to answer them.
Yeah, Matt, it looks like we do have some questions in the chat.
All right.
Let's see, the first question is, how long does the viral replication process take within the cell?
So it depends on the virus, but in the order of hours, and not many hours.
Okay.
So,
then,
yeah.
Oh,
No, like a handful of hours or less.
okay.
So,
and then with the strains and virus mutation rates, let's see, could you increase the possible reinfection in immune individuals with the various strains, I guess, that are the variants?
yeah, absolutely. And the reason for that is, so I told you like the spike protein is like the key, and it's going to go into one of our receptors, which is like the lock. And so our antibodies bind to the spike protein to prevent that from happening. So when antibodies bind to something, they bind to a very, very specific three-dimensional shape. So what happens with those mutants, the fear is if a coronavirus mutates in such a way that the spike protein changes its three-dimensional shape in such a way that antibody doesn't bind to it anymore, then all those immune system cells are worthless now. So it's all about three-dimensional shapes binding with each other. So a lot of the variants so far, some of them have had an effect on how infectious something is and all that, but they haven't evolved to change their shape of the spike protein so the antibodies can't bind to them. That's the nightmare. But that has not happened yet. Is it possible? Absolutely.
Mm-hmm. And you mentioned earlier, like the HIV virus versus, let's say, the influenza virus or smallpox virus, where smallpox, I believe you get one vaccine and it's good for your life. The flu, I guess because of the variants every year, and HIV, there is no vaccine.
Yeah, so those are great. Those are three. Yeah, so let's talk about polio and then flu and then HIV. So there's three different types of polio and they don't mutate. And there's always, as far as we know, there's only ever been three types of polio. It doesn't mutate, doesn't change over time. It's a very stable virus. And so when we get vaccinated for all three types. The virus doesn't mutate, so we're good to go. Right now, in the middle is influenza. What influenza or flu does most of the time is this process called antigenic drift, which means over the course of a year, when new viruses are made, it's kind of sloppy. Like viruses are kind of sloppy when they replicate, and so it makes little mistakes or changes pretty frequently, not all the time, but kind of in a pretty predictable manner. And so there's two molecules on the surface of flu viruses. You always see, like in the news, like H1N1 and all that. The H stands for hemagglutinin and the N stands for neuraminidase. There's just different kinds of those two different molecules on the surface, like the H and the N. So if the H and the N are slowly mutating and slowly changing their three-dimensional shape, our immune system can kind of keep up, right? But every now and then it changes the shape of those quite a bit. That's called antigenic shift. And so now all of a sudden the antibodies we have, whether they're natural or from a vaccine, don't work anymore. So we got to start over. So flu is kind of slowly evolving most of the time and slowly changing its three-dimensional shape. So our immune system struggles, but can kind of keep up. So then there's HIV. HIV is a nightmare because HIV is extremely sloppy with how it replicates and makes new copies of itself. In an untreated HIV infection, if one HIV virus infects one cell, it's going to make about 1 billion new viruses in one day. And statistically, every single one of them are going to be different from each other. It's going to make a mistake every single time it makes a new virus. Now, that's going to mean a lot of them are non-functional and don't work, but that's okay, right? If you make 100 million of something and 80 million of them don't work, but 20 million of them do, you're still ahead, right? So HIV strategy is to replicate super sloppily and super quickly. So it may end up with a lot of viruses that aren't functional, but it's still going to have a lot that are. But the advantage to that system is you're mutating so fast, there's no way the immune system could keep up. If someone had an untreated HIV infection and we could make a perfect vaccine for the virus in their body today, we could give it to them and we could make it, but it wouldn't work because tomorrow the virus would be different. So it's about the speed of evolution. Even though viruses aren't alive, if you have a genome, you can evolve, right? So.
We've got another question from the audience. What is the favorite theory among virologists? How did COVID-19 virus come about?
That is $1 million question. And that's actually interesting because that question is getting away from science a little bit and going more into politics, right? So we know that the virus came from Wuhan, right? So did the virus come from the wet market, like a transfer from, animals to humans, or did it come from the Wuhan Institute of Virology, which is 9 miles away? Right. So the people who were involved in that up until like a month and a half ago, pretty much poo-hooed that it could have been probably accidentally released from a virology lab, and it was all about transfer from animals. But you notice in the popular news, that's really been backtracked on. The World Health Organization went with Chinese officials to Wuhan to answer that question, but they weren't allowed access to pretty much anything. And so when they delivered their report, the director of the World Health Organization actually said while it was being delivered that whether or not it came from a lab or whether it came from the wet market needed to be further investigated. The old director of the CDC says it's his opinion, but he thinks it came from the Institute of Virology. It was released accidentally. So am I saying either one of those happened? No. I'm saying that either are possible, and there's not enough evidence to support either one of those hypotheses at this point, but they're both possible.
So we've heard the news that, as you mentioned, how do scientists or researchers determine if it was released from a lab versus natural spread?
That's a really good question. And that's a really hard question to answer. There are ways. So like when I was in graduate school, we were working on vaccines, and part of it, we would take a pox virus and then we would take parts of HIV and combine them into a pox virus, right? Not the whole HIV virus, but just certain genes of it. So the idea was you would have a pox virus that you could infect someone with, which we did for, hundreds of years, literally, but it would also express a single HIV protein. So maybe you'd be immune to both. And that went into clinical trials. I'm not exactly sure where that's at. But the point is, to do that, you had to use some drugs to force it to happen. So that required some genetic manipulation that you could probably find if you really looked, because you would find DNA sequences in the virus that shouldn't be there. But that's a way you could do that, but that doesn't mean you can still engineer viruses without having to do that. The interesting thing about the Wuhan Institute of Virology is they're doing what's called gain of function studies. And so here's the thing: viruses, they're not alive, but they can evolve, and things that evolve change because their environment changes, right? So one thing you can do is, let's say I had a coronavirus out in the world that came from a bat, and I wanted to make it much, much better at infecting humans, right? So what I would do is this: I would take a mouse and I would engineer that mouse genetically to express and make human ACE enzymes. So that angiotensin converting enzyme, the lock I told you about, I would take a mouse and I would engineer that mouse, which would be easy to do. So the mouse makes the human ACE protein, and then I would, take the cells from that mouse and then, make a whole huge stock of them. So I would have a mouse cell that had human ACE proteins on it, right? And then I would take that virus from a bat and I would infect those cells with it. The only way the virus from the bat could infect the cells is if it could bind to the human ACE receptor. And what I would do is do that over and over and over, hundreds of times maybe. Maybe the first time I took the virus and infected those cells with it, just maybe a couple of them would be able to bind to that ACE and infect the cells. But then the only viruses that would replicate would be those. So I would take that tiny pool, throw everything else away, take a brand new little pool of mouse cells with the human ACE protein and infect those with the virus. And over time, if I did that over and over and over, the virus would mutate over time to get better and better and better at infecting human cells because it's binding to that human ACE. You would use mouse cells just because you're not going to do experiments on humans, but you can sure take another animal, put human proteins in it, and then do it, right? So they were doing stuff like that, and they're upfront about that, right? And so the goal of that is to figure out how coronaviruses, evolve and the mutations they're going to make to become more infectious to humans. And people can debate whether that's worth it or not, right? That's not really part of my expertise, I guess, but that system definitely works, and they were definitely doing that system, those type of gain of function experiments at the Wuhan Institute of Virology. So it's just a complicated thing, right? Because it's so intertwined with politics. It's no mystery to anyone that politics have been pretty ugly for a while, and so that's really colored the discussion, to say the very least. So I think in the end, like, to sum it up, would be it's entirely possible that it came from, an animal. That's a common thing. It's also entirely possible it, probably accidentally came from that lab. That lab's like 9 miles away from the wet market. That's the same location. And I don't have an opinion either way because I don't have enough data. So.
We have another question. How can the mRNA vaccine be used for cancer, as you say, when cancer is not a virus?
No, it's a delivery system, right? So what if there was, what if a cell became cancerous because it wasn't expressing a protein? I could make an mRNA for that protein, put it in those cells, and now it would. Right. And there's this whole other family of stuff that we didn't even mention at all. There's this whole other kind of field of biology that's called regulatory RNA. And basically you can use RNAs to either cause genes in a cell to be expressed or to stop them from being expressed. So we didn't talk about that at all. So it's not like a vaccine at all. It's just that technology to get an RNA inside of a human cell has huge implications besides just virology. Essentially, I can take an RNA, put it into a cancer cell, and the mechanism that's causing that cell to be cancerous, I can shut it down. That's the goal. So.
And is the Johnson & Johnson vaccine the same technology as Moderna devices?
No, it's completely different. The Johnson & Johnson is traditional, right? So it's actually, so Moderna and Pfizer are the mRNA vaccines, like the future, in my opinion. The Johnson & Johnson is kind of a traditional vaccine. What they did is take a different virus called an adenovirus, and they've disabled it in such a way that it can still infect cells, but it can't replicate, so you're not going to get sick or anything. And it's basically a delivery mechanism to teach the cell how to make the spike protein. But it's more that's not uncommon. That's been done for decades. So, and that's why it took longer. Partially.
And we'll take one more question. Since it's 6:30. Respect everyone. Why does the virus vary among individuals? Some folks die and others don't get very ill. Considering this is a normal virus.
That if I had to answer that question, you'd be awarding me a Nobel Prize, right? But there are some things we do know, right? So, like if you look in general, I told you there's those B cells and T cells, and it's their job to go out and look for things that aren't there, to look for non-self, right? That's the basis of everything for that vaccine. But here's the thing, like, I'm trying to think of a way to describe this. So my B cells and T cells are going to recognize different things than yours are, right? So we all have B cells and T cells. They're all capable of recognizing foreign things, but that doesn't mean that if the same foreign thing goes into me and you, that our immune system is going to recognize it equally well. So you and I could both be infected with the exact same organism, but my B cells and T cells aren't as good as recognizing it as yours are. So you're going to mount an immune response faster, right? So all the signs and symptoms of the disease, the damage it's going to cause, you're going to be able to deal with that faster than I am. That's one thing. But there's also, there's just all these there's these things that are linked with, having, a robust immune system that we really don't understand, like the ins and outs of. Like, for example, there's some pretty good evidence. It was really anecdotal at first with COVID, that people who are low in vitamin D are really susceptible to like bad outcomes with COVID. And they're actually doing some really good, double blind studies now to see if that's actually true. It seems to be, but it's like good, what I would consider good research is being done on that now. So if we're deprived of something, there seems to be a link, underlying pathologies, like why does high blood pressure lead to such a COVID outcome? We don't know why, but we know it does. Obesity, same thing. And there's a pretty good link between, exercising and the robustness of your immune system. So a lot of it is just like lifestyle, giving your body the tools that it needs to fight things. But then also in the end, it's just kind of random. Like we just have different repertoires of immune system cells that may not be equally adept at killing a single organism. So. And if there's more questions out there, people want to stick around, I don't mind at all. I don't have anywhere to be for a few minutes. So, okay. Also, it's Friday night, so if you have something to do, don't feel like you have to stay.
Well, we have the question, is it true that the virus is a link between having blood group A?
That, I don't think the evidence supports that very well. I've never seen good data that would back that up. I'm not saying it's not out there. I'm not saying that at all. That's one of those things that there's and that's something that's going to be fascinating as time passes. We're going to be able to really look at a lot of these things in a lot more detail with a lot more data and have more time to really work those out. I know that's, people say that. I've never seen good evidence to support it, but it could be true.
Okay. And part of the issue is how long a vaccine will last, even in the absence of spike protein evolution.
That's a question that only time will tell, right? So we know that so for example, those pox virus vaccines, people who were born pre-71-ish have the scar on their shoulder. Those we know last for at least 40 years in most people. They last for a very long time. But there's probably people who had the pox vaccination where it didn't last, maybe a month, but in general, that one lasts for a very long time, right? And then on the other hand, there's ones we have to get boosters of pretty regularly, right? And so the short cop-out answer to that is you have to test that. So we're just going to have the time pass and see how long it does. So it's not a very good answer, but it's like, it's an honest one though. Like you just don't know until you've done the experiment. So.
Okay. We do have another question. Just Spanish flu, there was no vaccine. And I guess it lasted for over 3 years. It disappeared, or did we just reach herd immunity to stop the Spanish flu?
So that's true. It lasted a couple of years, and there were like 3 distinct waves, and it was very complicated because I had World War I going on and all of that. So that's another question that's really hard to answer. It was a very strange virus. Most of the time, like infectious diseases, if you take like people, it's going to kill the really young people and the old people, and everyone in the middle is going to be like the people that are, that survive because they're the strongest. It was opposite with the 1918 flu. It kind of preferentially killed people like in their 20s, 30s, maybe early 40s, which was very, very unusual. But the people who survived those infections, like if they were infected with that exact same flu, they would be protected. But the thing is, flu just changes. So flu, had that, it was an H1N1, right? It was an H1N1 flu. And so because it's always evolving, it eventually evolved away from being like that super pathogenic, right? And you think about it from a virus point of view. It's always interesting to take the virus's point of view. What a virus wants to do is infect you and not kill you. Like if a virus kills you, you're a worthless host, right? So what it wants to do is infect you, like replicate and live for a very long time and shed off many, many infectious virions. So if you look at the most successful viruses, they're not really pathogenic. Like Ebola is kind of a dumb virus because it infects you and then it kills the person. It makes it hard for it to spread. So there's a certain logic to like a virus wanting to evolve away from being that pathogenic. Like a perfect virus would be HIV from its point of view. It infects somebody, has like 10, 15 years to replicate and spread before it really starts to really adversely affect its host. So that's a good question, but also not with a clear, really super cut answer. Most likely, it just kept evolving, that anthogenic drift process I told you about, and then eventually it assumed a form that wasn't quite so pathogenic.
I think we'll wrap things up for insight. And if you're interested in learning more about the class that Dr. Pearcy teaches at Yavapai College and to enroll, please visit www.yc.edu. That's for Yavapai College. And in the next day or so, look out for an email asking you to complete a short survey. Your feedback is very important and will help us determine what you'd like to see in future science cafes. So right now we're planning our next event for the June-July timeframe. So look out for an email invitation. Friend us on Facebook at Verde Valley STREAM Council. And please invite your family and friends. And as always, the more the merrier. And thank you all for joining us for now. And good night. Hope to see you all very soon.
Happy Friday. Thanks for coming, everybody.
Bye.